everyone. I really appreciate the opportunity to share our work with you and to learn with you all. I'm really privileged to be at the Broad Institute, where I work directly with the researchers who are conducting the primary analyses that are discovering the genes and the variants that are contributing to autism. And so the title of my talk is Supporting Diverse Needs in the Autism Community Using Genetics, and I'll be building upon some of the work that the previous speakers have been talking about. Wanted to just note that the Autism Science Foundation did partially fund this research through my fellowship, but they didn't influence any of the study designs, results, or decisions to publish, and I myself have no financial disclosures related to this. So I'd like to start by noting that I'll be using the descriptors people with autism, autistic people, and people on the spectrum, as I know of folks, interchangeably, as I know of folks who use these descriptors. And I think these very descriptors really speak to the tremendous diversity of preferences and experiences in the autism community. And I'd like to take a minute to highlight this phenotypic diversity. There are people with autism who cannot communicate their needs in ways that others can consistently understand, who require assistance in basic self-care activities like using the toilet, and who oftentimes may react in ways that can harm themselves or the folks around them. And folks would need 24/7 supervision for the duration of their lifetimes. And there are also autistic people who can fluently communicate their experiences, independently adapt to the demands of everyday life, and take care of their families while thriving in their careers. But at the same time, they can also experience significant challenges as well, as there are higher rates of ADHD, anxiety, and depression, as well as suicide among people with autism diagnosis compared to people without. And because different people can have very different needs from each other, I think that the kinds of services that they could benefit from are also likely to differ from each other. And we're starting to recognize that for some people, their difficulties may be attributable to genetic differences. And there's fewer groups of people in which genetic differences are more abundant than in the autism community. So my roadmap for this talk is to describe how genetics can help support diverse needs in the autism community. And specifically, I'll share some of our recent research into phenotypic diversity, which are individual differences in phenotypes such as traits, developmental histories, and diagnostic descriptors, its relationships with genetic diversity or individual differences in DNA sequences, which you've started to hear a little bit more about from Dr. Jeste, and ways we are translating this knowledge towards developing individually tailored resources and hopefully services down the line. And the goals of my research are really to try to provide the community with more opportunities to thrive in the ways precisely that they'd like to thrive. And I hope that by the end of this talk, you have learned some of the ways in which genetics can help inform these opportunities. So as alluded to earlier, one of the most important ways in which genetics can be useful to the autism community is clinical genetic testing. And while clinical genetic testing is recommended by several professional medical associations for all US children with autism diagnoses, most of these children have not yet undergone first-line genetic testing. And I think this is a missed opportunity, as a genetic diagnosis can help autistic people and their families understand the behavioral, developmental, and medical challenges that a person has been experiencing. It can help clarify expectations for what the person may need in the future and help them access more tailored supports like developmental resources, medical recommendations, and family support groups and parent advocacy groups. So for example, and I'm sure many of you know, a genetic diagnosis of Phelan-McDermid syndrome can help explain why a child has been experiencing symptoms like vomiting or seizures or problems with urinating. And it can point to getting their gut and health and kidneys checked regularly. So given the potential utility of genetic testing, I think it can be helpful to understand which genetic differences can be diagnosed in autistic people. So we often call genetic differences genetic variants, And genetic variation associated with autism is extremely diverse, and it can range from common to rare. So common variants associated with autism are inherited. They're found in under 1% of the general population, so they can be found also in folks without neurodevelopmental disabilities, and in fact, probably all of us carry a few one way or another. And individually, they have very, very small effects on phenotypes. In contrast, rare variants associated with autism are often non-inherited. They're observed in fewer than 1% of people, and actually many people with autism diagnoses don't necessarily carry a rare variants. And individually, they can have very large effects on phenotypes. So of these rare variants, we're particularly interested in what we call neurodevelopmental disability associated variants, or NDAVs for short. So NDAVs are non-inherited, and they can arise from a change in a single DNA letter that affects one gene to hundreds of DNA letters that can affect multiple genes. And the genes that they affect are genes that are associated with neurodevelopmental disabilities like autism or intellectual disability. And we know that variants in these genes can significantly impact brain development. And currently, many NDAVs can be returned as a genetic diagnosis through clinical genetic testing. So we recently studied how carrying an impactful rare variant like an NDAV is related to when autistic children start learning the necessary skills for independent functioning. And this horizontal line represents the first 7 years of life from left to right, and here are the ranges of ages at which most autistic children who do not carry NDAVs attain developmental milestones like walking, speaking words and speaking phrases, and toilet training. And in comparison, children who carry variants like NDAVs, which are shown below, they show on average delays and greater variability in attaining these developmental milestones. So based on this work and the work of other research groups, we know that multiple phenotypes in autistic children are associated with carrying rare variants like NDAVs. So using these phenotypes, we asked, can we estimate how likely a child is to carry an NDAV and therefore to receive a neurodevelopmental genetic diagnosis? To answer this question, we analyzed the DNA sequences and a lot of phenotypic information from over 3,000 children with autism diagnoses. And some of them were in the Simon Simplex collection or in SPARC. So thank you to all those who have participated in those studies. We studied how different combinations of phenotypes may be associated with different probabilities of carrying an NDAV, which we call a genetic diagnosis probability. And for people without autism diagnoses, the average probability of receiving a neurodevelopmental genetic diagnosis is about 1%. Among people with autism diagnoses, however, the average probability goes up to about 7 to 10%. But of course, we know that not every autistic child is going to have this average probability. It's very unlikely they'll all have the same probability. And it turns out we can be more specific than that when we look at the diversity of phenotypes a child can have. So we found that genetic diagnosis probabilities vary substantially depending on the combination of phenotypes autistic children have. So for example, along with other phenotypic information, male autistic children who don't have a congenital heart defect, who started walking by the time they were a year old, so not delayed at all, and had a cognitive score above 110, their genetic diagnosis probability is right around 1 to 3%, which really isn't that different from the average genetic diagnosis probability for people without autism diagnoses. But in contrast, in female autistic children with congenital heart defect who started walking 2.5 years, which is pretty delayed, and had a cognitive score below 70, which is on par with intellectual disability, their genetic diagnosis probabilities can range from 12 to 76%, and which is very, very high and is much higher than the probabilities, the average probabilities among children with autism, which was, as I said before, 7 to 10%. And there's so much diversity in these probabilities among autistic children, which really speaks to me the importance of considering these individual differences in the research we conduct, as well as the resources we develop. So here we found a way to estimate an autistic child's range of genetic diagnosis probabilities based on easily accessible information about the child. And I think these personalized probabilities can really help clinicians and families prepare them for what they might expect when the child undergoes genetic testing. So we've been translating this research into a public web-based suite of resources that families and clinicians can use. And to build these resources with and for the autism community, we consulted multiple advisory groups of people with expertise and lived experience. So this included genetic counselors, bioethicists, autistic adults, and communications experts. And we also worked with the Autism Science Foundation to recruit small focus groups of parents and pediatricians to gain their feedback about prototypes of this resource. And really hearing from and integrating all of these diverse perspectives was really an invaluable learning experience for all of us on the development team. And we're calling this resource Minerva, and we're excited to launch it in the coming months. And Minerva is designed to help clinicians and families of children with autism learn about genetic diversity in autism and to integrate this information into considerations around clinical genetic testing. And it's really one of the first genetics education resources that was, one, tailored to autism's unique genetic architecture, and two, designed with and for autism community members. So using Minerva, you can learn updated scientifically supported information about genetic diversity in the autism community. You can then choose to enter an autistic child's information anonymously, and it won't be saved, so that they can estimate the child's range of genetic diagnosis probabilities. And then you can explore several resources for seeking clinical genetic testing and supporting people with genetic diagnoses. And we really understand that clinical genetic testing is a highly personal decision for every family. And what we hope that Minerva can do is just provide more information and empower families and clinicians to have these important conversations and to make informed decisions together. So Minerva is just one example of how we can translate genetics research into a tailored set of resources. But there's so much more to do. We're recognizing spectrums in the autism community not only at the phenotypic level, but at the genetic level. And ultimately, where I think we need to go as a field is to integrate information from all types of genetic diversity to gain a more complete picture. So over the next few years, my collaborators and I plan to study rare and common variants together and to examine how these genetic factors interact with environmental and developmental factors among autistic people. And we're really interested in seeing how these factors may jointly influence phenotypes that are key to people's quality of life, such as their capacity to thrive in different environments, given the challenges they report facing, and their benefits and risks that they may experience from using different services. And this research, I think, could help lay the foundation for designing clinical trials that can help build more tailored services that can be recommended for more people on the spectrum. So in summary, we now have some of the tools and opportunities to understand both phenotypic and genetic diversity among autistic people, and to start using that understanding to help connect autistic people to resources like clinical genetic testing, which in turn can lead to more tailored services. And one of the first things we made progress on in that journey is to link phenotypes with NDABs to help estimate a child's genetic diagnosis probabilities, and to integrate this into Minerva, which families and clinicians will ultimately be able to use. We've made a lot of progress, but of course there are many other factors that affect the challenges autistic people may face and the services they may need. And I think one step to getting closer to that is to study how phenotypic diversity is associated with genetic factors as well as other important factors like environmental and developmental factors. And because with this information, we can then start to design clinical trials that could inform more personalized, effective services for autistic people and their families. And this is all towards giving people on the spectrum more opportunities to thrive precisely in the ways they'd like to thrive. And with that, I would like to thank all my collaborators, our funders, the participants in the studies, and to you all for your attention. Thank you.